In this unit, we're going to be talking about something called the cell cycle and how it relates to cancer. Now, the big question that we want to answer is how, do cells divide, and how does this division ultimately lead to cancer? But we have to take a step back. We had to first figure out how we go from here to here to here. So what I want you to do, I want you to write down at least 3 steps relating to cells between the picture on the left, progressing all the way to the picture on the right. Now, hopefully, in one of your 3 steps, you mentioned something called fertilization. Now, if you didn't, fertilization is where a sperm and an egg come together and form something called a zygote, and that fertilized egg then goes through cell division over and over and over and over again. Now, this cell division gives rise to identical cells. However, the cells and their functions eventually will vary, and that is a process called differentiation. So, in a bigger context, differentiation means doing things on an individual basis or doing things kind of differently than everyone else around you. In this context, differentiation means that each cell undergoes a process so that it has its own special structures and functions. Right? So a skin cell is different than say a hair cell. And so these specialized cells come together to form different kind of levels or different levels within the hierarchy of life. And we've talked about this already this year. And so cells form together to become tissues. Tissues form together to become organs. And organs come together to form organ systems. So let's go a little bit more in depth for understanding different hierarchy of life. Now again, we have talked about this year, but this is just a quick review because we're going to be talking about these different levels a lot. So on the very top of our hierarchy, we have something called an organism. Right? That's one that might be you or me. It's one person within a species. That organism is made up of a number of organ systems. So each organ system has a number of organs within it. So for example, the digestive system is made up of your stomach, your small intestine, your large intestine, your gallbladder, your colon. So each one of those individual organs come together to form an organ system. Now you're going to see a pattern here pretty quick. Each one of our organs has several different tissues working together, all kind of working towards a common goal. For example, in your stomach, you have the outer lining that has a function, an inner lining that has a function. Ultimately, there's a number of different tissues that are all working together to do everything that a stomach has to do with digestion and breaking down foods. Continuing to move forward, obviously we have tissue. So tissue is a number of identical cells working together for a common goal, right? So our muscles have muscle tissue. That tissue is made only of muscle cells. For example, our stomach, right? The outer lining of our stomach might have a specific cell that makes up that tissue. And so going down to our most basic unit with a cell, our cells have our 6 characteristics of life, which we have talked about. And there are countless different examples of cells. Muscle cells, skin cells, hair cells. There's a very long list of cells within our body. So again, this hierarchy needs to be understood when we talk about the development of human bodies and the division of cells. So here's the big caveat to all of this. For the most part, differentiation is irreversible. So what I mean by that is once a cell is given the information to become a skin cell, for the life of that cell, it's going to be a skin cell until it dies. There are very few ways around that. This is why our brain cells are so important, because it takes a long time for them to divide. And so if we lose brain cells, we really can't get them back. Also, if we break our back and cells die as a result, and we lose the ability to use our lower extremities, right now there's really no way to reverse that and lead to more cell growth in the areas that were damaged. But there are a couple different areas where science is starting to experiment with to potentially lead to us being able to use undifferentiated cells. So stem cells are cells that have yet to be differentiated. They've yet to become their kind of final goal. They have the ability to become multiple types of specialized cells. So there are 2 primary types. Type 1, embryonic stem cells. These are cells that have never been differentiated. They're kind of waiting in line to get their job or their task. They all, if they were allowed to move forward, would eventually get a specific function, though. The second type is called adult stem cells. So these stem cells are found in bone marrow. So if you don't know what bone marrow is, it's the kind of the inner core. It's kind of a membranous substance within our bone, and here lies our stem cells. Now it can't become any cell. It can only become bone, blood, cartilage, fat, or connective tissue cells. But it's still considered a stem cell because it can become multiple different types of cells and tissues depending on what the body needs. Now, we have something called the cell cycle. The cell cycle is the kind of cornerstone, the keystone of this unit. Now, the first part that we need to understand is that this is the cell division that occurs in eukaryotic cells. And it ultimately happens over and over and over again. So the 2 primary purposes are growth and repair of cells. And there are 3 main phases. Interphase, this is where the cells grow. This is by far the longest stage. It takes the most amount of time. Then we have mitosis, which we'll talk about very soon. And cytokinesis, this is where the cells actually start to divide and become their own cells. And this happens at the end of mitosis. So, we're going to give a quick overview of the cell cycle. And really we're going to focus right now on the first primary stage. So again, interphase is where the cell grows. Ultimately, cells, just like our bodies, grow as they age. And there are three parts to this stage. The first one, G1 or gap 1 phase, this is where the cell grows and makes proteins. The S phase is where the chromosomes replicate. And then the G2 or the gap 2 phase, it goes similar to the G1 phase. The cells grow and make more proteins. So ultimately, at the end of interphase, because of the S phase, each cell has 2 sets of chromosomes. And this is going to set us up as we move forward. So, as a reminder, a chromosome is one long thread of DNA with a bunch of genes. Now remember, genes, which are kind of coding information for specific proteins and other things, genes make up our DNA, and our DNA is wound tightly together to form a chromosome. And again, remember, this is for organizational and space kind of purposes within the nucleus. Each of our human body cells has 46 chromosomes, and that's unique to us. Other animals have more or fewer chromosomes. And for a normal cell, each chromosome comes in a pair. So obviously if cells are mutated or something went awry, this could change, and it would potentially have pretty significant impacts on the cell. Now, before we really get into it, there's a couple of vocabulary words that I'm going to be using a lot that I want to make sure that you know. The first one is called chromatid, and so a chromatid is 1/2 of a duplicated chromosome. So if you look to the right, half of that kind of X, the orange X, would be a chromatid. Now, when you're talking about both chromatids, you call it sister chromatids. Each kind of line of that X is exactly the same. They are identical. So the 2 right there, those would be considered sister chromatids. Next, we have the centromere. This is the region right in the middle where it looks like the chromosomes come together, and this is where the chromosomes are really condensed. And the last one is going to be telomere. So telomere, these are the ends of the DNA molecules. So the very tips of the chromosomes, those are called telomeres. Okay, that was a ton of information, but that was all preparation simply to help the cell divide. So this long time period within interphase is all set up to get the cell ready and prepared to divide. And that is what we're going to be moving on to next.